Hi everyone, welcome to week 3 SendForC's video on photosynthesis and biochemistry. My name is Jessica. I'm a 4th year molecular and cell biology major.
And my name is John Riley. I am a fourth year biology major.
So this is like a brief overview of what we're going to be going over today. Number 1 is photosynthesis, and number 2 is cellular respiration. And then we're going to go into biochemistry and a little bit about biochemical techniques. And then throughout the video, we're going to have concept checks to test your understanding.
All right. Yeah, once again, my name is Riley. I'm going to talk to you guys real quickly about photosynthesis. So the very basics of photosynthesis, as you know, is that it's a process that plants use to produce energy and food. Very basically, plants take in carbon dioxide, light, and water to produce glucose and oxygen. Very specifically, the chemical formula for this process is it's going to take up 6 molecules of carbon dioxide and 6 molecules of H2O to produce glucose and 6 molecules of oxygen. The most important thing about this process is that it takes place in the chloroplast. And if you remember from our previous mini lectures, we have talked about plant cells before and all of its organelles. Now, an overview of chloroplast is that this is the site where photosynthesis occurs. This contains chlorophyll, which is the molecule that reflects green light but absorbs all other colors, hence why plants appear green. Chloroplasts contain thylakoids, which are the green disc structures that you see on the inside, and sacs of those thylakoids are called granums. These are also the sites where light-dependent reactions of photosynthesis occurs. Now, we're going to do a little overview of photosynthesis. There are 2 types of reactions in photosynthesis, light-dependent reactions and light-independent reactions. Light-dependent reactions consist of photosystem 1 and photosystem 2. Now, the purpose of light-dependent reactions is to convert light and water into chemical energy and to release oxygen. Light-independent reactions consist of the Calvin cycle, and the purpose of that is to convert carbon dioxide into glucose. To dig a little deeper, light-dependent reactions, as mentioned before, consist of photosystem 2 and photosystem 1. Sunlight energy is absorbed, water enters in and is broken down. This produces oxygen, and the hydrogen ions are then transported across the membrane. Now, some of those hydrogen ions are used to convert ADP into ATP, and some of those are used to convert NADP plus into NADPH. Now, this is just what photosystem 2 and 1 looks like a little bit. It may look a little daunting, but we're going to go ahead and dive in a little bit deeper and explain a little bit more. Now, photosystem 2 occurs first, despite its name. So the first step is that sunlight is absorbed into chlorophyll molecules, and an electron from that chlorophyll molecule is going to absorb that energy and it becomes excited, and this process is called photoexcitation. The electron then leaves the chlorophyll molecule and awaits its next step. Chlorophyll now needs a new electron because it just lost one from excitation. This is where water comes in. On step 3, water comes in and it splits. One hydrogen ion is now replenished in the chlorophyll and the oxygen molecules are now released. The other hydrogen ion is used to make ATP. This leads us to the 4th step. The excited electron from step 2 gets carried through the electron transport chain to another chlorophyll molecule. And this leads us to photosystem 1. The 5th step is that sunlight is absorbed once again, and a new electron gets excited and awaits its next step. Now, if you remember that same electron from steps 2 and 4 that just got carried through the electron transport chain, that will replace the excited electron that the new chlorophyll just lost on photosystem 1. The recently excited electron gets carried through another enzyme to NADP+. The last step is that NADP+ is combined with the hydrogen ions and the excited electron to make NADP+. NADPH. All right, now we just finished light-dependent reactions. The newly synthesized ATP and NADPH will be used in the light-independent reactions. These also may look a little bit daunting, but don't worry, we'll go ahead and explain it. There are three main steps to the Calvin cycle, which is what the light-independent reaction consists of. The first step here is called carbon fixation, and we can see here that Rubisco will help bind 3 carbon dioxide molecules to 3 RUBP molecules or ribulose biphosphate. This breaks the long chain into 2 parts to form 6 3-PGAs or phosphoglycerates. The next step is called reduction, where 6 ATP molecules put phosphate groups onto the ends of the 6 3-PGAs. An additional 6 NADPHs puts an electron onto the other ends of those same 3-PGAs. Continuing on, this now forms 6 G3P molecules or glyceraldehyde 3-phosphates. However, it requires 5 G3Ps to recycle and remake RUBP, so this just leaves 1 G3P molecule to be used to make glucose. The third and last step is called regeneration. The 5 G3Ps are now converted back into the original 3 RUBPs through the use of ATP. All right. Now here are just a few concept check questions that we're going to go ahead and ask you to do on your own time. The first question is going to ask true or false. A stack of thylakoids, which are located in the chloroplast, is called a Rubisco. Second question, part A, is what are the names of the 2 complexes in light-dependent reactions? Part B is which of the 2 occurs first? And the last question is what is the name of the molecule that is the starting and end product of the Calvin cycle?
All right, thank you, Riley. So we're just gonna move on to cellular respiration. My name is Jessica, in case you forgot. So our cells need energy to carry out important biological processes and grow. So we mainly get our energy in the form of ATP, which is adenosine triphosphate, and this is the structure of it right here. So there's 3 phosphate groups and this adenosine molecule here, and this is attached through a phosphodiester bond. And so the reason we can't just make ATP and it's floating around in our cells is because it's high energy and pretty unstable. So it's typically inside the cell and stays inside the mitochondria. So glucose and other molecules are transported and broken down in our bodies. And this process is catabolism. And this is how we, get our energy production. And this schematic on the right side just shows you theoretically the yield that we should get from all of these metabolic processes in our bodies that theoretically would give us 36 ATP overall. And yeah, so this is just from one glucose molecule, and this is basically the process that we're going to explain and go into. So glycolysis occurs in aerobic conditions, which means it requires oxygen, and it's the breakdown of glucose into pyruvate. So this is an energy dependent process, so it requires some ATP to get started, but it does produce ATP. And this is a spontaneous process with the use of energy coupling and enzymes that are involved. Since it is ATP dependent, that's what helps it be spontaneous. Just know that the reverse of this would be non-spontaneous. It's gluconeogenesis, which is creating glucose, and that also requires a different process to make it spontaneous. The Krebs cycle, also known as the citric acid cycle, converts the pyruvate to acetyl coenzyme A, and this gets transported into the mitochondria, which is commonly known as the powerhouse of the cell, because that is where most of the ATP production is in our cells. There's a series of reactions, and it releases carbon dioxide and GDP, which gets converted to ATP for energy use. So actually this cycle produces GDP, which is a different form of ATP that indirectly produces ATP. And lastly, I'm just going to explain lactic acid fermentation, which is what happens in our bodies when we are in anaerobic conditions and when we have excess pyruvate. It's going to cycle into this cycle here. And there's a bunch of enzymes that basically convert pyruvate to lactate, and this can get exported out of our cells and used for other metabolic processes as a carbon source for energy. So this is just a very general overview, and you will learn more in detail about this if you decide to study biology. It's very interesting to know how our cells are working. So I'm just going to give a brief introduction to biochemistry and the two aspects of biochemistry. Structural essentially explains how enzymes, substrates, proteins, and other molecules interact with each other, and they bind to each other. And this is, spatial orientation determines a lot of the functions in our bodies and in our cells. And you can study these interactions using different types of techniques. Microscopy, you can stain these molecules with fluorescent dyes and do different tests to measure consumption and their interactions. Amino acids and DNA structure is really important for these protein interactions because they are the building blocks of our DNA and of these proteins. And metabolic biochemistry is essentially what the last slide was demonstrating on the use of enzymes and different metabolic cycles to explain all of these physiological processes that occur. So both of these have different processes in plants, animals, and eukaryotes. So it's very interesting to just know that the general idea, and especially for humans, this is how we make a lot of scientific advancement and medicine and other fields of biology. So this is a really good example to understand how reactions are occurring in our bodies. And further, you're going to learn these reactions in more detail for these different metabolic cycles. So enzymes, increase the rate of reaction by lowering activation energy, but they do not get consumed in the reaction. So they are recycled, essentially. They go into the reaction and they should come out, and they are able to go into another reaction and do the same process over again. So it's important to know that enzymes do not alter the equilibrium constant of reactions, but they only increase the rate. So they essentially don't not shift reactions one way or another. And for biochemistry, it's important to understand the concept of spontaneity, which refers to the tendency of a reaction to occur without external input or work. And this is important because a lot of reactions in our bodies cannot occur without proper enzymes and reaction coupling. And we will come back to this idea when we look at the electron transport chain. So this schematic down here is just showing exergonic versus endergonic reactions. You also can refer to them as exothermic or endothermic, but essentially exergonic reactions refer to energy releasing, while endergonic refers to energy absorbing. So if a reaction is exergonic, it will occur spontaneously, but that doesn't really tell us much about the rate, but that's just something to consider. So when we look at the electron transport chain, which is inside the mitochondria, and this is where a lot of our ATP is made, how did they figure out the electron transport chain was creating ATP? Well, Peter Mitchell proposed this hypothesis that ATP production is coupled or linked to oxygen consumption. So they confirmed this through testing these complexes by adding this substrate here called succinate and measuring oxygen consumption over time. And then they also did a separate experiment to measure ATP production in. There is another complex here that's ATP synthase, and that is the complex that uses these protons and this proton gradient to essentially create ATP. And this oxygen consumption is directly linked to ATP synthesis. So that's what this graph is showing here, that once you add all of these reactants or reagents, now you have increased oxygen consumption and increased ATP synthesis, and they are linked. And when you inhibit one process, you essentially can inhibit the other process from occurring. You can have oxygen consumption without ATP production, but that's a little bit more complicated. But all you need to know is that essentially these processes are linked, and that is called the chemiosmotic hypothesis. And this is just a research article that I wanted to show you from 2018, so it's pretty recent. So even though we, study this a lot, it's, we're still figuring out how oxidative phosphorylation and all of this reactions going on in our cells are occurring and how they can affect even cancer therapy. So this is just very interesting to look at if you are interested. And to sum it up, we have some concept questions here to test your understanding, and you can answer this after the video. Is glycolysis a spontaneous process? Why are enzymes important? What happens to cells in anaerobic conditions when they need energy? And how can we study energy production? And you can answer all these by referring back to the slides in the video.